Hi, welcome everyone. Happy Friday. Woohoo. Today, Jörg Pfannmöller, yes, is giving a presentation. He's part of the Biomedical group, and he's here now 2 years, almost 2 years. And he will tell you a little bit more about the simulation of the functional MRI, basically. So enjoy.
So hello. Yeah, so basically we really, or the idea of my project is really to simulate fMRI. So that is something that has not yet been achieved with a arbitrary high degree of realism. So we always, that there are already good simulations, but they always use certain approximations. So one very common approximation is this, more or less this functional hyperaemia that you see here in the title. And functional hyperaemia means that you only care about the changes in the blood flow, so and in the vascularization. So actually, what you finally would like to do, you would like to connect fMRI, so this imaging technique, with the processes in the neurons. That is possible. The processes in the neurons are coupled to the processes in the blood, and fMRI measures the changes in the blood. It is maybe possible, but currently we are at the stage that we take our knowledge about what's going on in the vascularization of the brain. It's all about the brain. What's going on in the blood, the changes in the blood that are connected to that, and we use that as our basis. So, and the question that we can answer with that is how specific this blood signal is, how specific towards the side of the neurons. And an important question that has up to now not yet been answered is how specific fMRI is along the entire cortical depth. So we already have simulations that tell us something about the upper layers, but we don't know about the entire cortical depth. And on the first slides, I would like to introduce why that is important before I get to the details of the simulations. So how does an fMRI experiment look? I mean, it's already done for 20 years. So the typical fMRI experiment is that you have a scanner. Today you even got high field scanners like 7T and 9.4T for humans. And so to say, in the classical version of fMRI, you did your experiment in the scanner, and your result looked somehow as you see it on the right side here. So you got what you got out of the experiments were these blobs, which basically signal the relatively large regions in the brain that are active during your experiment. And today we know that fMRI is for sure capable of measuring or of investigating the large-scale structure of the brain. That is what you see here. And now currently there's a lot of research going on if we can make the next step so that we get to smaller units of the brain that should be investigated in the next generation of experiments. So, and as I said before, fMRI is a blood signal. So we actually would like to investigate the activity of the neurons, but fMRI measures changes in the brain. And this figure here illustrates how the vascularization in the cortex looks. This is the visual cortex of a monkey. And what you see here is that the vascularization, the vessels are have a structure that is easily to see. Here in blue, you see the veins. In red, the arteries. The gray part here in the middle are the capillaries. And the blood flow is simple. The blood comes in through the arteries on the surface of the cortex, what you see here. Then the diving arteries, so to say, transported into the cortical gray matter. then the blood flows through the capillaries towards the counterpart of the diving arteries, the diving veins. The diving veins collect the blood in the cortex, transport it to the cortical surface, to the PL surface veins that you see here. You can even see it better around here. And then the blood goes back to the heart and the lungs, and so on. Okay, and so today we know a lot about the structure of the red vessels and the blue vessels, but the structure of the green vessels is not so well known. So there are still a lot of open questions, especially concerning humans. But they are especially interesting if you would like to image what's going on in columns and in layers or along the cortical depth, because in the capillaries happens the oxygen exchange, and the capillaries are closest to the site of neurons. And so to say, the simulations should help to should also help to increase our understanding of how specific the signal of the capillaries, the signal from the capillaries actually is, how specific it is to the activity of the neurons, not only to the site of the neurons. Okay, so right now there is still one thing missing. So this year in this, my investigations are mainly about the bold contrast. That is the mostly applied contrast. It is mostly in most applications measured with gradient echo sequences. So probably most of you here, if you do fMRI, most of you will use gradient echo sequences. And these sequences have from the application point a lot of advantages. So often you can't use anything else, but they also have let's say some particularities that you have to know about if you want to get a high specificity towards the capillaries. That can be illustrated if you use here this oversimplified bold curve and this inset from the vascularization. So the advantage of gradient echo sequences is that they allow you to have small voxels within a short measurement time. So with other techniques that gets a little bit more difficult, and you need small voxels if you only want to measure the gray matter. Yeah, that's simple. And if you want to measure something like layers or columns, you even need smaller voxels. So the cortical gray matter is about 2 millimeters in humans, and you definitely need sub-millimeter voxels if you only want to measure gray matter. Now, why is that important? That is important because these gradient echo sequences get a very large contribution from the veins, not only from the capillaries. From these veins here, these diving veins and these surface veins. And you definitely want to get rid of the surface veins. So you can achieve, and that is something that you can achieve if you have small voxels, if you just measure here in that region. Okay, so far so good. Now let's assume you have that, then you can apply masking and you are free of the signal from the PL veins, but you still have the signal from these ascending veins. And this is especially bad if you want to measure cortical layers, because the blood flow in the veins causes an additional problem. The blood flow in the veins couples the different layers. And and that means small voxels don't suffice if you want to get rid of the vein signal, if you really just want to have the capillary signal. And you can get, but there's a trick, and you can get rid of that vein signal if you just measure in a very early time point. So in this curve here, in this bold curve, basically this large peak here is the vein signal, but if you measure here, you get the capillary signal. And this is, so to say, currently the experimental setup to get closer to the signal of the capillaries. And that's very nice. That already made it already possible to measure columns. And an important prerequisite for that is a high field scanner. So you definitely need a 7T scanner or higher to achieve that in humans. Okay, and this is, so to say, the current state of the art in high field fMRI. That is applicable even if you're not like the world's most leading expert. But now, if it is about layered fMRI, there are still a lot of very fundamental questions. So you can do columnar fMRI if you really press on it. Concerning layered fMRI, the question is still today more or less if the capillaries themselves are specific enough to allow you to get to the level of layers. So the point is, if the blood flow of if the region that is perfused is much greater than your active layer, regardless what you do, then you will never get to the point that you get a signal that is layer specific. And so the simulations the work on the simulations that I do, and the work and the work in other projects that are connected to it, target largely on this question. So now, besides the if you want to do those simulations, you need what you need as a basis is a very precise knowledge of the anatomical structure of the vessels, and that is something which is, and the work on these networks is currently in progress. There are just a few networks that allow you to investigate this question on the specificity of fMRI towards layers across the entire cortical depth. There are not even any for humans right now. So right now, the only networks that we have are from mice. So in this figure, one of them is shown here, and currently we have exactly 4 of them. These networks are measured using microscopy, and that is, so to say, another branch which is connected to this question. And these, so to say, this is done from researchers that are very good into, for example, into photon microscopy. Since we have them, we can in principle do these simulations with much larger bands than in the prototype simulations from 2015. So to say, at this point, my project starts. And a huge challenge today is something which I didn't expect, if I'm honest. In the beginning, I thought I would work on a very different thing. So a huge challenge today that is not solved. Even the fundamental physiology is not known to a degree that we can easily do it. Is the computation of the blood flow in these vascular networks. And it is really so that even the world's experts, the best people around, currently know very well that our knowledge is rather limited and that we have to live with approximations, and we have to figure out, so to say, the approximations that work. Now, and I don't know if I should go too much into detail here. So what we basically do is we work with something that is similar to what you do if you compute the current in a resistor system. So in an electric current, we replace the network by straight networks by straight segments, and use the Hagen-Poiseuille law that is visualized here. Maybe you know it. Maybe it already crossed your path. And then one very important physiologic effect that I never heard of. So before I worked in that project, I did fMRI for several years. But I never heard of the most fundamental effect concerning blood flow. That is the so-called Faraos effect that is visualized here. And that just says that if you have a tube like a vessel, and you have a fluid like blood that consists of, let's say, a true fluid like the blood plasma and red blood cells, and you make that liquid flow through the tube, then the flow organizes itself as you see it here. It organizes itself so that the red blood cells are so to say concentrated in the center of the tube. So I don't know if anybody of you ever heard of that. For me, that was a huge surprise because nobody even mentioned it in all that time. And what we basically do in our experiments, we go one step further. We apply an approximation. We use the Hagen-Poiseuille law and this Faraos effect. And we approximate the blood as a fluid that flows only in a part in this smaller part of the vessels that we have in our networks. Okay, and right now I'm in my project concerned with figuring out the right configuration to make everything run in a large network that covers the entire cortical depth and parts of the white matter. So that's not so simple. I got to include some further effects, but I don't want to go into any details around here. However, I worked with the previous version, and I would like to show all the other steps that are necessary to get finally to the bold signal, to a bold signal out of those simulations. These are shown here. So in addition to the flow, you also gotta compute convection, that is mass transport in the blood vessels and diffusion to the tissue. Then you have various other steps. After you included this convection, you can compute the susceptibility distribution within the vessels. And if you go through all of that, finally you get to the bold curve. And it is so that basically what I found up to now is that if you include the latest knowledge on the blood flow, suddenly the interpretation of it could even be possible that the interpretation of the bold effect in these specific that this may lead to a change in the interpretation of the bold effect in the early phase, where it's highly specific to capillaries. And yeah, with this, I would like to close my talk and thank you for your attention.